Okay. Hello, everyone. It's a pleasure to introduce Valentina Perosa for our seminar series. Valentina is an MD PhD with expertise in advanced neuroimaging and computational modeling of vascular contributions to Alzheimer's disease. Her main research goal is to understand the mechanisms that link cerebral small vessel disease and Alzheimer's disease to identify targets for therapeutic interventions. During her postdoctoral training at MGH, she combined in vivo MRI, ultra-high resolution ex vivo MRI, histopathology, and deep learning techniques to investigate the neuropathological correlates of MRI-defined vascular lesions. This research led to impactful results in publications in ACTA Neuropathological and Annals of Neurology. And she's the recipient of many awards, including the Alzheimer's Association Research Fellowship and a K99 R00 award. Valentina is currently an instructor at MGH, and today she will be talking about the interaction between microvascular and AD-related pathology in the medial temporal lobe.
Thank you so much. Okay, definitely don't have anything more to add to this biography. Thank you. Thank you so much for having me here and for the invitation. So yeah, as Maria said, I'm going to talk about the interaction between microvascular pathologies and AD-related pathologies, which is, I guess, one of my favorite topics. So to start, first of all, I will give some background on the topic and just hint on some previous work that we have done, and then explain a little bit more about the links between small vessel disease and AD-related pathologies in the medial temporal lobe, and then dive into the sex differences in the cross-links between small vessel disease and AD-related pathologies, also in the medial temporal lobe in the same cohort, and then just outline some slightly different studies and future steps that we've been working on. So, oh, that's weird.
Although I can see it.
Yeah.
Can you move forward in that way? Okay, I think just that person.
Okay. Everything good?
All right, that's good.
Yeah. All right. Then I'm gonna continue. So yeah, we know, of course, that there is a contribution of vascular disease to Alzheimer's disease. So this is something that has been really the focus of research for quite a while now, and there is a lot of evidence, like no matter from what angle one looks at this topic. So first of all, of course, we know that many patients that have been diagnosed with Alzheimer's disease, in fact, don't only have the defining pathologies of AD, such as tau tangles and amyloid beta plaques, but we can also find in their brains cerebral amyloid angiopathy, for instance, Lewy bodies and phospho-TDP-43, which is another pathology that coexists often with Alzheimer's disease. So that's one point, but also there have been some studies, and I'm just going to cite this one, which had inspired me back in the days, which is a study that has been done on the ADNI cohort, in which they saw that the markers of vascular dysfunction actually preceded even markers of amyloid beta accumulation in the tentative progression of Alzheimer's disease. So that's something that is quite remarkable in my opinion. And maybe even more striking is also the evidence that comes from epidemiological studies. So, for example, we have seen that the vascular risk factors have been connected to dementia, but also that in both in developed countries or like in high income countries and but also in low and middle income countries, the incidence of dementia has decreased comparably to the better control of vascular risk factors. So that's also very strong evidence, but also dementia has been directly related to obesity and to hypertension and to diabetes, and also to hypertension has been related really to the accumulation of amyloid beta in the brain in mid-age. So this is all, in my opinion, quite strong evidence for this interaction. But so I have to be more precise when I talk about Alzheimer's disease-related pathologies in this study specifically. I'm talking about the defining pathologies of AD, so amyloid beta plaques, neurodegeneration, and tau tangles, but also about phospho-tau DNA binding protein 43 and its accumulation in the cytoplasma of the cells, which is another pathology that has been related to Alzheimer's disease and also has an independent effect on cognitive decline in patients. And when I talk about microvascular pathology and small vessel disease, in this case, I'm talking about cerebral amyloid angiopathy, which is the accumulation of amyloid beta in the vessel wall, and also, especially in the leptomeningeal and the cortical arteries, and also about arteriolosclerosis, which is defined by the thickening of the blood vessel wall with high linosis, loss of smooth muscle cells, and just dysmorphic changes of the vessel. So this is what I mean when I mentioned them. And in the diagnosis of small vessel disease during life, we rely especially on neuroimaging markers of cerebral small vessel disease. And because, of course, we cannot see the vessel directly during life, they're too small to be imaged most of the times, at least in clinical practice. So what we look at are downstream markers of small vessel disease. And this can be non-hemorrhagic markers, such as the enlargement of perivascular spaces that you can see, especially in T2 weighted MRI, but then also white matter hyperintensity in the FLAIR. And then on the other hand, also hemorrhagic markers of disease, such as microbleeds, corticosuperficial siderosis, and bigger hemorrhages, like ICH. So, and one thing that I always love to mention about in this context is actually the idea that small vessel disease was related to Alzheimer's disease is definitely not news. There was some pathologists that already looked into that. And well, my favorite is Gaetano Perosini because he comes from my hometown, Udine. But he was a colleague of Alois Alzheimer, and he actually noticed straight away that there was CAA in the brains that had been diagnosed with AD. And he already thought that there might be some sort of interaction there, but then he decided to join World War I, and he died on the battlefield. So there was this strain of research who was kind of dead for quite a while. But in any case, despite all these premises, 100 years later, we still don't know whether small vessel disease and AD-related pathology co-occur in the brain, just randomly, let's say so, because of age, for example, or if they have a cumulative effect on cognitive impairment independent from each other, or whether there is a causal interaction between the two of them. So this, and of course, it's very hard to have interventional studies or experimental studies in humans. Interventional studies take a very long time, and they're also, of course, expensive and full of confounding factors. And therefore, one way that potentially can help to disentangle this is to use human brain pathology. And that this has been done for this purpose by a couple of studies, but not a ton. One, for example, found that CAA, patients with CAA had a higher chance to be staged at a higher BRCA stage when all other factors were equal. Another one also found that CAA, when it was moderate or severe, led to higher tau burden or was associated to higher tau burden in the brain. And finally, there has been quite a few studies actually that looked into the interaction between phospho-TDP-43 pathology and arteriolosclerosis. So specifically, this one showed that arteriolosclerosis in the posterior watershed regions of the brain was associated to higher burden of a phospho-TDP-43. Whether that's because of hypoperfusion, we don't know. Anyways, but there is an interaction there. And there is one region of the brain that, in my opinion, really deserves particular attention while tackling these links, and that's the medial temporal lobe. Of course, we know the MTL has in it also the hippocampus, the entorhinal cortex, the parahippocampal gyrus. So all these structures that are incredibly important for global cognition and for memory. And not only that, but the MTL is also the region of the brain in which a lot of these pathologies that I've mentioned occur altogether. So we have tau tangles that appear very early on in the process of Alzheimer's disease. At some point, also amyloid beta plaques join, and phospho-TDP-43 is predominantly actually located in the medial temporal lobe. And finally, small vessel disease is also highly prevalent here, even if not as severe as maybe in other regions. So I've been interested in this topic now for quite a while. And for example, there was one older study which we had conducted when I was still in Germany, in which we tried to look at the vascularization of the hippocampus using 7 Tesla time of flight angiography, so high resolution time of flight angiography, which could in this set of studies, we could visualize for the first time the arteries that directly supplied the hippocampus. And we confirmed pathological studies and showed that the supply of the hippocampus was either by both the anterior and the posterior region of the brain, so by the anterior choroidal artery and by the posterior cerebral artery, or only by the posterior cerebral artery. So there were this, roughly speaking, these two variations. And what we saw was that patients that had at least one doubly supplied hippocampus had better cognition and a better verbal memory, and also there was better hippocampal integrity when using BBM on these high resolution scans. But another point was that this was particularly relevant for patients who had also small vessel disease. So that kind of the single supply was a worse risk factor for worse cognition in case of small vessel disease. So when I joined the Stroke Research Center at MGH, I kind of also one of my lines of studies kept being interested in the hippocampus, and what we did was to investigate the relationship between hippocampal integrity and the cognition in a CA cohort, and we saw, frankly, not really surprisingly, that there was a relationship between the loss in integrity in all the hippocampal subfields and all the cognitive domains, also in the CA patients. And maybe more interesting, what we saw was also that PSMD, which is a measure of white matter integrity, was the only marker of small vessel disease that was related with the decrease in volume in the hippocampus. But still, we don't know what is the relationship between small vessel disease and AD-related pathology in the medial temporal lobe of patients with Alzheimer's disease. And as I was mentioning, there is really not that many studies on that, actually none, at least not to my knowledge. So we went back and took a cohort of patients from the Alzheimer's disease research, from the Massachusetts Alzheimer's Disease Research Center, and we tried to investigate this neuropathological signatures of the interaction between small vessel disease and AD-related pathology. And this one is just one example of one of these cases in which you can see how all the pathologies that I've mentioned before coexist in the same brain, in the same hippocampus. So arteriolosclerosis is here in this LHE stain section, which stands for Luxul blue with hematoxylin and eosine, and we will see more of this later. So yeah, as I was mentioning, we went in and looked at this cohort, and we collected a total of 157 AD cases. And there are unfortunately not a ton of clinical information about these cases, but we could at least also place them along the continuum of the severity of Alzheimer's disease. And we used Brack stage as a proxy for that. So we grouped the cases according to the Brack stage group, at least. We diminished the group so to make things easier. So and we also knew whether they had dementia or not. And for each case, what the ADRC does is to have a standardized sampling of the brains, right? So we have standardized sections that are cut and stained for several markers. And the ones that we were interested in this case, were the ones that contain the amygdala and the entorhinal cortex on the one hand, and the hippocampus and the parahippocampal gyrus on the other. So these two blocks were the ones that we looked at. And I think one of the strengths of this work, in my opinion, is that we used deep learning-based quantitative measures of pathology in order to quantify the hallmarks of disease in a more objective manner. So instead of saying, oh, there is a lot of amyloid beta, there is little amyloid beta plaques, so we tried to really quantify them in order also to have a more complex statistical model so that can be analyzed later. And this is just a quick schema about how this quantification works. So first of all, of course, the sections need to be prepared and stained. We then digitize them with a high resolution microscope, which is called the nanozoomer. And they are then uploaded on the Euphoria Cloud, which is a proprietary software that we use then to annotate about 10% of the whole cohort. This 10% is representative of the variability in the sections that we see throughout the cohort. We annotate them, and then we train the AI model using the software over and over and over again until we're happy with it. And once we're happy, we apply the model to the entire cohort. And of course, so there is also a validation process, first of all, quality control, of course, but also an external validation. So some expert pathologists or some trained research personnel looks at them and annotates the sections as opposed to the AI. And finally, we export these measures and use them for statistical analysis. Another advantage of this approach is that it can also be used towards other cohorts. So the model does not disappear. It can also be applied to other cohorts. And in fact, that's actually what we did for the amyloid beta model, which is this model trained on amyloid beta sections, which really distinguishes between cerebral amyloid angiopathy on the one hand and amyloid beta plaques on the other. This model had actually been trained on a completely different cohort, and we could implement it on this cohort quite easily. And a similar observation was also made for tau tangles model, for the tau tangles model. So this one recognizes tau tangles, and in this way we have at the end the density of tau tangles in the region of interest. And the same is true for the phospho-TDP43 model. In this one, we can also see the density of phospho-TDP43 cytoplasmatic inclusions. So the only model that unfortunately we were not able to train for was for arteriolosclerosis, because there is too much heterogeneity and there is not an actual marker for arteriolosclerosis. So we just look at the vessels and we can grade them from 0 to 3 according to their severity, and 3 is the most severe. You can maybe here recognize how there is no more smooth muscle cells, the vessel is thickened and also kind of dysmorphic, so that tells us that something is wrong there. So all these pathologies were assessed in 4 different regions of interest that we manually segmented. So amygdala, entorhinal cortex, hippocampus, and posterior parahippocampal gyrus. And in pink, you can see where we assessed arteriolosclerosis, because it's to be found especially in the white matter. So we looked at also at the white matter of the parahippocampal gyrus and of the entorhinal cortex. And we had, as I mentioned, validation measures that were very good for the models, but also I did a sanity check and controlled whether the tau tangles density, for example, was consistent with the Bragg stage and actually increased with the Bragg stage, which was the case in all regions. And I did the same also for the amyloid beta plaques percentage area. So this also was consistent with tau stage groups. But then, as I was mentioning, one of the goals was to show what the burden of small vessel disease across AD pathology was. And we could see that CAA was mostly more severe in more advanced Bragg stages. And on the other hand, this was not true for arteriolosclerosis, which kind of remained the same, or at least it was not significantly more severe in more advanced Brach stages. But one further aim of the study was, of course, to look at the interaction between small vessel disease and other pathologies. And in fact, what we saw was that CAA was a predictor of amyloid beta plaques percentage area throughout all the regions of interest, and also irrespective of Brach stage. So it was an independent predictor, or was independently associated, better to say, with amyloid beta plaques. And the same was true also for tau tangles density. And we could also see that the CAA predicted how the density of tau tangles was in these regions of the brain, and especially in the entorhinal cortex and in the parahippocampal gyrus. This was not the case for phosphatidylP43, which seemed to fall out of this line. And we also did not find any relationship between arteriolosclerosis and these pathologies, unfortunately. I was hoping for it. But then another aspect that we investigated in this cohort were also enlarged perivascular spaces. And I don't know how familiar you all are with what an enlarged perivascular space is, but perivascular spaces are this compartment that are around the blood vessels. And they have been implicated in brain clearance. So brain clearance is, of course, this process that is supposed to clear out some of the noxious proteins out of the brain. And the perivascular spaces seem to be a very important compartment for that. And another thing that has been postulated is that their enlargement is related to brain clearance dysfunction. So in other words, brain clearance is actually one of the mechanisms that we think might plausibly explain the relationship between small vessel disease and the accumulation of AD-related pathologies. So that's why we find this aspect so interesting. And in a previous study that I led together with my mentor, Suzanne van Veluwe, what we look at was the enlargement of perivascular spaces and their relationship with other pathologies in CAA brains. And what we found was that the enlargement of perivascular spaces in the white matter was strictly related to the accumulation of CAA in the overlying cortex. So in other words, we think that the PVS enlargement is possibly a result of a blockage in clearance that occurs in the upstream portion of the same vessel. And this is like, the study is a little bit more complex than that, but I don't have time to go into the details, but there are multiple aspects within the study that pointed towards that direction. And therefore, we decided to look at enlarged perivascular spaces also in the medial temporal lobe. And in fact, in this case, however, in the medial temporal lobe, one of the things that we observe is that within the enlarged perivascular spaces that you can see here in white in this LHE stained sections, we often see very sick and unhappy arteriolosclerotic vessels that are thickened and deprived of any smooth muscle cells and probably not really super functional anymore. And in order to also quantify the perivascular spaces, so we used a semi-automatic method implemented in ImageJ that segments the perivascular spaces and calculates the percentage area of the EPVS within the white matter. And we then observed that arteriolosclerosis was not a predictor of AD-related pathologies, but it was a predictor of the enlargement of perivascular spaces. And not only that, actually the model, the linear mixed effect model that best explained this relationship was one that included cortical CAA as an interactive factor in it. So it is possible that an interaction between CAA and arteriolosclerosis severity leads to an enlargement of perivascular spaces and thus that the interaction between these two small vessel disease pathologies leads to clearance dysfunction. This is, of course, all possibilities that what we can do with this pathological data. and it all needs to be better investigated, maybe in vivo in mice or in other ways, but this is what we can see here. And what is also interesting is that we found exactly the same result in a completely different cohort and in cortical regions of the brain, and not only in the medial temporal lobe. So this was actually a cohort of CAA patients that had this. So to conclude this part, CAA seems to be an independent predictor, or has an independent association with the burden of amyloid beta plaques in the medial temporal lobe, and it also has an association with the density of tau tangles. And this is not true for arteriolosclerosis, and it does not have a relationship with any parenchymal AD-related pathologies, but arteriolosclerosis seems to interact with CAA in predicting the burden of enlarged perivascular spaces. And phospho-TDP-43 in this cohort didn't have a relationship with small vessel disease or with enlarged perivascular spaces, nothing. So arteriolosclerosis has been also puzzling me for quite a while. I think it's my new favorite topic in general, and because we don't really know what it's doing. We have no good way of quantifying it. So we see this and this thickened vessels, but there is not a specific marker as opposed to CAA. We don't have a specific marker for arteriolosclerosis. And also there seems to be quite a bit of heterogeneity in the pathology in itself. So we don't really know what is happening there. But one thing that we did look at and found out in another study was that arteriolosclerosis can kind of mimic CAA sometimes. And it does not only exist in the very deep areas of the brain, but also in the subcortical white matter. And in fact, what we did was to target with ex vivo MRI, we targeted microbleeds, so this microhemorrhages that I was talking about before. And we then use this pathology to go after this microbleeds and find out, okay, which pathology is in the vessel that broke down to provoke this cerebral microbleed. And what we saw here was that it was actually arteriolosclerosis and not a CAA. We couldn't see any CAA in these cases. These are, of course, not the majority, but it needs to be taken into account also for potentially in the future for clinical purposes. But yeah, and then after this little digression, another thing that we looked at in the medial temporal lobe of this Alzheimer's disease cases was sex differences in pathology, because we know, for example, that females have a higher chance of getting Alzheimer's disease. The prevalence of Alzheimer's disease is higher in females than it is in males. Also, females accumulate more tau pathology than males, unfortunately, and faster, and it seems to have even a stronger effect on cognition. And furthermore, we are more vulnerable against vascular pathologies, especially arteriolosclerosis is more prominent, more prevalent in females than in males. On the other hand, CAA seems to be more severe in males than in females. So plenty of reasons to look at sex differences in AD. And one final one was also that the differences between sexes seem to be mediated by ApoE4 genotype, and the ApoE genotype is, of course, the most proven and most important genetic risk factor for Alzheimer's disease. So, and here I have to thank Francesco, who is looking at the analysis of this cohort. Francesco is a research fellow at the Stroke Research Center, and by analyzing this cohort in this respect, he found that there was a difference in arteriolosclerosis. So females had indeed more arteriolosclerosis and less cortical CAA in the medial temporal lobe. So that's very consistent with previous studies. Another thing that he saw was actually less, this is a little bit inconsistent with previous studies, but he saw a little bit less amyloid beta plaques in females as opposed to males, but he did see more, he did see also more tau tangle density. And but finally, I think what is actually most interesting about this part of the study is that we had ApoE genotype for 66 of the participants. And what we saw here was that there was a positive interactive effect between the presence of one ApoE4 allele in the cohort and the fact of being female. So those two factors together led to a higher chance of a high tau tangle density in the medial temporal lobe. And that's also consistent with previous literature, and it's independent of the presence of arteriolosclerosis or of CAA. And it's nice that, now we have been genotyping the entire cohort. So I think Francesca's analyzing the data this week, and we'll find out whether that holds true for the whole group. So to conclude, CAA severity is higher in male than in females. Arteriolosclerosis, on the other hand, is higher in females. Female sex is an independent predictor of higher density of tau tangles and lower amyloid beta burden in the medial temporal lobe. And the interaction between female sex and presence of ApoE4 allele had a positive effect on tau tangles density. So limitations is that we don't have a lot of clinical data, especially we don't have data for menopausal onset age or status, and also we have some missing ApoE. So then for the very final, how am I doing on time? I'm not, I cannot see. Okay, good. Okay, good. I hope I'm not talking too fast, sorry. But then maybe allow me to switch gears and go into another area that I have been interested in in the past few years, and that's the use of quantitative susceptibility mapping to investigate several aspects of small vessel disease. So I don't know how many of you are familiar with what QSM is, but it's a processing method that allows us to see and quantify susceptibility in the brain throughout the whole brain. So this is something that we have worked with quite a bit in Magdeburg in Germany, where I studied. And what happens is that there is a whole post-processing method that I'm not going to go into, but it combines in a multi-equus T2 star weighted sequence, it combines the magnitude and the phase images in order to achieve this final map that you can see here, which gives us in each voxel and in each region of interest the actual quantitative value for susceptibility in terms of parts per million. So that's what QSM is. And one of the most important applications in this case is, of course, that it's possible to differentiate iron, or in general, also paramagnetic substances in the brain from the susceptibility artifacts that are also provoked by diamagnetic substances in the brain, such as calcium. And because we are dealing with a lot of hemorrhagic markers in small vessel disease, this can be particularly relevant for CSVD. And in fact, one thing that we saw was that QSM was able to have like a better sensitivity towards microbleeds than other T2 star weighted sequences. I mean, of course, here we're looking at 3 Tesla versus 7 Tesla. So don't get me wrong, I know that's in any case, there is a difference there. But we could see that also in 2 7 Tesla sets of data. And maybe most importantly, what QSM is able to do is to differentiate between calcifications that look like microbleeds on T2 star weighted sequences and actual microbleeds. And because the QSM for the classification is hypointense instead of hyperintense. So we can do that. And QSM, however, has also been invoked as a potential measure of blood-brain barrier leakage, because it can potentially measure the leakage of iron products from the blood, from the vessels in the surrounding tissue, in the perivascular tissue. And of course, we know, I mean, blood-brain barrier leakage is one very important step in the remodeling of the vessels towards a pathological state. Potentially, we think it might be one of the first steps. And therefore, we have an incentive in trying to quantify this in vivo. And there was one study that looked at QSM and compared it to DC MRI, which is a kind of MRI that uses contrast agent to actually quantify blood-brain barrier leakage, and that's has been there is pretty solid evidence that that's reliable, and QSM was related to this DC MRI. So I got intrigued into this idea. And another study that I found very interesting was this study that also looked at QSM, but this time ex vivo, so in brain, formalin fixed brain samples. And they found that QSM was related had the susceptibility artifacts that were related to iron enriched microglia around white matter hyperintensities. And this was in MS patients. And then last but not least, blood-brain barrier leakage, especially in the medial temporal lobe, has been evoked as a very an important marker of disease in Alzheimer's disease, even though some of this evidence has been disputed. That's also something that one needs to mention. But especially this paper by Montagne is very interesting because they found that in DC MRI, blood-brain barrier leakage in the hippocampus was related to presence of ApoE4 genotype and also with cognition. So we thought it would be interesting to validate QSM as a measure of blood-brain barrier leakage, but using ex vivo tissue. So like measure QSM ex vivo and then validate it as a marker. And furthermore, also because there has been some controversy, we thought it's interesting to assess blood-brain barrier leakage in the medial temporal lobe across different and pathologically confirmed BRAC stages. And this is something that we're doing in brains with AD pathology. So what we do is to cut a block from the medial temporal lobe from formalin fixed brains, and then we put it in a little falcon tube, and we use fluorinert, which is a viscous liquid that avoid the presence of bubbles, and then scan these samples using a custom-made solenoid coil at 7 Tesla MRI. And this allows us to have a very high resolution of about 100 micrometers isotropic, which is nice. Very beautiful images. And what we can then do is like to take the same block of tissue and embed it in paraffin and cut it in serial sections and look for markers of blood-brain barrier leakage in the histopathology. One of these is fibrin extravasation, as you can see in this image, but also we are looking at, of course, iron and calcium to see. And related then to the reconstruction of the QSM that derives from this multi-echo sequence. And here I have to thank Lydia Marazzo, who is a very, very talented undergraduate student who has been working with me recently, and she's really trying to match the ex vivo MRI of the hippocampus with the histopathology, and which you could just see. And this is another example of histopathology, even if it's a bit of a crappy section, but stained for fiber. And so she has been doing a very amazing job. And there is another step that we have been using for this particular samples is also micro CT, which you can see in pink. So the micro CT allows us to have an even higher resolution than the MRI. And it's done on the paraffin embedded block, so that it's an ideal in-between step between the MRI and the registration of the histological sections, which is otherwise quite hard because of the deformation through cutting, etc. So what we hope to do with this is to really look at the single vessels pathology and look both at what the QSM signal is around it and then compare it to the histopathology on the one hand, but we also want to do more than that and really look at single vessel pathology as related to down or upstream AD-related pathology. So that's going to, of course, work in progress. And with this, I think I'm also done. So I just want to thank you all for the attention and the whole from the Lobe Lab, which is amazing, and many other people for helping out. Thank you.
Super interesting.
Any questions?
Yeah. I think,
Oh, sorry.
go ahead, yeah.
Yeah.
Your last few slides there. So when you're comparing MRI with micro CT and then histopathology, what are you looking across those three modalities? Like what information do you look into those?
Yeah, so the micro CT is like a one last slightly crazy idea I had recently, to be honest. But what we it was we are experimenting with it, but what the advantages of the micro CT is that it allows it to register the sections, so the histological sections, much better using the software, because the micro CT's image when the block is already in the paraffin, so embedded in the paraffin, which means it's like stuck there, in other words, you know. And on the other hand, for the MRI, the block is a little bit larger, and then we have to cut it so that it can be embedded and sectioned. So the micro CT is kind of an in-between step for that, first thing. And secondly, the resolution is much higher. So we have here 11 micrometer isotropic resolution, which means that we can really look at a single vessel. And yeah, MRI can also do that. There is at the NIH, they actually do that, and I hope that we can collaborate with them at some point, but it's takes even longer so, to image. So yeah. Did I answer your question?
Yeah. So it's more like the first two are structural, and then like the last one is also structural, but just like the higher resolution from the IHC, basically.
Yeah, so the last one is tells us what is there, basically, in terms of pathology.
Okay.
Exactly. So the last one tells us there is fibrin, which means there is blood-brain barrier leakage, or there is iron, which is consistent with what we see in the QSM, for example. That's what we hope to find out.
Okay.
Yeah.
Thank you.
I have a question. I think you have to help me understand what you think is going on in terms of the mechanisms. I think the first set of studies was really interesting because I almost by the end thought, oh, so what's actually happening is there's all this vascular pathology that is worsening the brain, and then you've got this, you know, at some point, it sort of becomes a cumulative burden of
Yeah.
vascular pathology plus this amyloid plaques and so forth. And that's where you end up with the Braak stage 4 patients where everybody looks so similar in terms of the CA versus amyloid. Then where does but then I got confused with the blood-brain barrier leakage thing. I was like, wait, how does all this fit into, because that's a different thing going on. And I'm just wondering if they all tie together or these are sort of independent things.
So I think, yes, I think that's what's going on in the interaction between small vessel disease and the AD-related pathology. So I think that's good. I guess you got the message that I was trying to convey. And the blood-brain barrier leakage, it's actually a marker of vascular dysfunction. And it's a very early marker of vascular dysfunction, so which we can potentially measure in vivo. So we assume that there could be blood-brain barrier leakage already in vessels that don't show really the pathology in terms of CAA or arteriosclerosis. So my colleague, actually, Mary Kotzberg, who is an attendee at MGH, she did a study in which she kind of defined the progression of what happens in the vessel in itself while the pathology develops. And from what we could see, most likely the blood-brain barrier leakage is the first step that goes wrong. So that's where it fits in.
Collateral damage, but early sign of
Yeah.
something is going wrong because the system is not normal.
Exactly.
It's
So
not the consequence of
Yeah.
all of this.
no, it's not. It's actually it's a marker of early vascular dysfunction. that's what we think it is.
So can I just ask a follow-up?
Sure.
This is, at the end of, by the time you get to this patients where you show that each of that CAA and the other measure, forget, not arteriosclerosis, but CAA predicts not just, I'm trying to remember your slides, AAA, the amyloid burden, but also the functional-
tangles.
Yeah, and the tau.
Yeah.
Is it just like the brain at that point is like just in atrophy, nothing can stop it, and it just sort of like a rolling stone, everything is getting worse? Or the thing that stops me from just saying that's probably what's happening,
Yeah.
because now you're saying there's not that much relationship between arteriosclerosis in some cases, but other times it seems to be a marker. It also seems to be a factor. But why is this just not the brain just getting worse in all ways?
I mean, I think in this set of data, at least, what speaks against that is the fact that we corrected for Bragg stage. So this association that we see is actually independent of the Bragg stage.
The association
Between CAA and amyloid beta plaques on the one side, and between CAA and the density of tau tangles on the other side. So it doesn't, that should not, like we included that, which is again one of the reasons why I love using this deep learning based measures of pathology, because in this way we could use more complex statistical models. So we use this linear mixed effect models in which we can actually correct for things like that, and that's nice.
It's not just that it's not the case that everything is just getting worse.
Yeah. I don't think so. I think it's irrespective of that. I mean, obviously, it's an association as everything, right? So we don't know whether it's causation, and we might move forward also with more like inference-based analysis, or at least that's something that I would like to do at some point when I stop writing grants. Yeah.
Yeah, I think we have a
But,
question online.
Oh,
Do you want to read it? Do you see it, or
wait, just one
I can
second.
read it for you?
I'm curious, how early can you identify broadband barrier leakage, and what are the exact biomarkers that are measured? I assume you mean in, I don't know if it's true, but I assume you mean in Alzheimer's disease, maybe. So in Alzheimer's disease, it seems to be a very early marker of AD. For example, this study by Montaigne, they saw that it was there even before cognitive decline started, and it was even there in cognitively unimpaired patients who had one ApoE4 allele. So it was, it's an early marker of vascular dysfunction. And the exact biomarkers, well, it depends. So if we look in vivo, we have especially contrast agent-based MRI, such as DCE MRI, so we can see the extravasation, and this measure is called the K-trans. And if we look ex vivo at the pathology, one is the especially the fibrin extravasation and fibrin accumulation in the within the vessel and in the perivascular area, and at the same time also iron and or I hope to see some iron, but I'm not sure about that yet, and even IgG. We can stain for IgG and also see accumulation of that in this pathology. You're welcome.
With your ex vivo data, we also have like
No, I wish so much, but I am looking whether how many of those, like just simply a matter of time, but I really want to see whether they also have at least some ex vivo MRI, because I'm sure some of them must have some ex vivo MRI and some of them must have even in vivo MRI. I just like didn't get around to it yet, but yeah.
One.
Yeah.
Okay, I was wondering if you think there would be like an interaction between like CAA severity and like the density of the tangles and like the progression of the disease. Like,
yeah, that's a very good question. I mean, this is kind of the next question that one needs to ask, right? But the problem, of course, with pathology, with brain pathology, is that it's inherently cross-sectional. Like, there is no way to, although I think the Allen Institute in Seattle, they modeled the tentative progression, like using machine learning, they modeled the tentative progression of AD in the pathology using many cases. So I'd be super interested in looking into that, but I think I'm not sure that pathology is the right method to answer that question in that sense. Yeah.
And just out of curiosity,
Yeah.
are there studies using QSM longitudinally? So you say that blood-brain barrier leakage is one of the first things that happens. So, you know, if you localize where there's leakage,
Yeah.
does that essentially predict where people will have some of these, you know, biomarkers that were more
Potentially.
Yeah.
Yeah. Not that I knew of with this specific question, because this idea of using QSM as a blood-brain barrier leakage marker is, I don't think it's very widespread and it's not validated. So that's why we're doing this kind of work. So I don't know. I know that there is a center in Munich in Germany. They have a high frequency study on CAA in the CADASIL patients, and I think they are also interested in QSM, but I don't think as a marker of blood-brain barrier leakage. But let's see, maybe I convinced them to let's see what comes out of this, and then whether it's worth pursuing. Yeah.
So going back to that MRI in the last few slides, are you interested in doing kind of like looking at in a reverse direction and make the biomarker for the clinic? So like do like 7 Tesla of the patient and then try to diagnose if they have arteriosclerosis, CAA.
Sorry, can you repeat the question again?
So we were showing the multiple
Yeah.
the 7 Tesla and then the micro-CT.
Yeah.
You were saying you want to be looking at like the structural changes in MRI and then also the markers within the immunohistochemistry.
Mhm.
Are you interested in creating like a clinical diagnostic?
Oh, yeah, well, ideally that's the case, right? Like the reason why ex vivo MRI has fascinated me, and the reason that literally the reason that I came to Boston for was because it can serve as a bridge between what we see in the MRI, which we think we see something, right? But then through ex vivo MRI, the positive thing is that you can validate it pathologically. But then also the opposite is true, that if we define a marker, a neuroimaging marker in ex vivo MRI, we can maybe at least partially use it then in vivo to assess the same thing. Exactly. Yeah, that's the idea. Should probably state that a little bit more clearly.
Very cool.
Oh,
Very
okay.
cool. Thank you.
You're welcome.
You don't see any questions online?
No, I think we're good.
Sounds amazing. Thank you so much.
Thank you for having me. That's great.